Hello, AP biologists! Today I'm wearing my Star Wars shirt, so it's going to be a good day. Here I have a plant. I brought a plant in because I thought it was a good way to show that this plant is actually made of several other plants, but they all started out from an original parent plant here. And then through mitosis or asexual reproduction, these other plants grew because they inherited the chromosomes from that original parent plant through the process of mitosis. So that's one way you can inherit DNA into a new organism. Another way you can inherit DNA is through sexual reproduction or meiosis, which is what you covered in your last module. So an example of this would be like my family. So this is my great-grandmother. This is a really old picture. This is my grandma. This is my mom, and this is me, right? So this is a lineage. And some of the characteristics we share because we inherited chromosomes down through generations, and some are different because there's variation like crossing over, and also fertilization from having dads and things like that, so that we don't all look the same. And so that is an example of inheritance as well. So today, though, we're going to talk about gene linkage, which is my favorite subject when it comes to inheritance. So we have a problem here about gene linkage, and then we'll go over it quickly. We'll get started with this FRQ question. It looks like we're talking about chromosomes. You can read the prompt first, is always a skill, to see what you're looking for. So two genes were genetically linked. Okay, so linked, I immediately know that means they're on the same chromosome. So that is what linked means. It's always good to define words. Describe how the proportions of phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a GGDD individual. Okay, now I need to refer to my prompt because I don't know what the F1 is. In certain species of plant, there's a diploid number of chromosomes. Flower color is controlled by a single gene in which the green allele is dominant and the purple allele is recessive. I'm going to definitely want to make a note of that. Shout out to Alfonso for always making notes and annotating his prompts. It's a great skill. So G equals dominant and G equals recessive. So if I have to look back on this, I don't have to read. Plant height is controlled by the gene where the dwarf allele is dominant. So capital D is dwarf, means short, and tall allele is recessive. So I'm going to write those down as well. Great, now I'm all set for that. The P generation, the parental generation, is the homozygous or true breeding of two different types of plants, so of a green dwarf plant and of a tall purple plant. So that's my P generation. They were crossed to produce my F1 progeny. So if I were to cross those, what would I get? Well, remember, you inherit one allele from your mom and one allele from your dad. And in the case of Mendelian genetics, we assume that these are, if they were not linked, you would inherit, again, one capital G from your mom, and then you'd inherit a lowercase g from your dad, and then you'd also inherit a capital D from your mom and a lowercase d from your dad, right? Because that's the only options of what you can inherit. So you are going to be heterozygous. I know that you said that because you're so smart. So the F1 generation would be heterozygous because you are getting one allele from each parent. Great. So now I know what my F1 progeny is. So I'm crossing an F1 individual with a homozygous recessive individual. Great. So I'm just going to draw that cross. We quick jot it down so that way I can see what I'm looking for. Now remember, this is a dihybrid cross, these are linked. So, but what would it be like if it was not linked? So we just want to quick think about, like, what would that look like? So I don't have to FOIL these gametes. They're always going to be the same because you would inherit a little g and a little d no matter what. However, the heterozygote gametes do need to be FOILed because you could inherit them differently due to independent assortment. So you could get a capital G and a capital D. You could get a capital G and a lowercase d. Or you could do the first outside. You could do the inside, lowercase g, capital D. And then last, lowercase g, lowercase d. Okay, So whenever I see this, I immediately think to myself, like, okay, well, it's going to be the same all the way down here. So the only difference is across the top, which means I'm going to have a 1 to 1 to 1 to 1 ratio. So my ratio should be 1 to 1 to 1. Great. So I have a 1 to 1 ratio. If these were independently assorted, so if I got one allele from each parent. However, they're not. They're linked. So when things are linked, I should know that that means that I'm going to look like my parents. So describe how the proportions of phenotypes. Most of the phenotypes will look like the parents. And I'm going to state what that is: green and dwarf, or tall and purple. A few of the offspring will look different than the parents. So example, green and tall, because of crossing over. So again, I'm saying most of my phenotypes are going to look like my parents because the genes are linked. So I've described what I would see happening. So I'm just going to check my answers to see if that is correct. And in fact, it does. It says that a majority or greater than 50% would have the parental plant phenotypes. And I said most would look like the parents. So I have gotten a 1 out of 1 on that. So I'm going to go back to here and give my score 1. Yes, I've passed AP Biology. Just kidding, if only it were that simple. A couple of notes I put in the slides. Another slide about gene linkage, since I think it's something we really struggled on when that was in play. Just knowing that gene linkage just implies that if genes are linked, then most of the offspring look at like the parents because you inherit those chromosomes together. And the only reason that you don't look like your parents when genes are linked is because of crossing over, like what we saw in the meiosis do now from last Thursday, when you were crossing over your parts of your gene or your homologous chromosomes were swapping genes so that you could get fruit flies that look different than their parents. If genes are on separate chromosomes, though, then we inherit them separately, like the laws of independent assortment, and then you get offspring in that typical 1 to 1 to 1 to 1 ratio with the probability of offspring. Another quick note that I just didn't cover as much as I should have, we talked about the endosymbiotic theory, which stated that like old eukaryotes used to envelop bacteria, and that's how we got the mitochondria. So the mitochondria used to be a bacteria that used to perform aerobic respiration that lived its own life, and then one day it was eaten by a larger cell, and then the cell just kept reproducing it over and over again. And same with the chloroplast. It used to be just old prokaryote, but then was enveloped and the cell continued to make it. The reason we think this is the case is because there is DNA in mitochondria, and that can only really happen if it was a prokaryote, not just a regular organelle. So what does that mean? If my mitochondria has DNA, how do I inherit that DNA? And I just want to note that DNA from the mitochondria or the chloroplast comes from your mom or the egg. Like whenever I ask you a question about where does this DNA come from and you don't know, it's like not obvious, usually it's from the mom. So your egg, the egg provides that mitochondrial DNA, and that's just something that you should know. Well, I hope that you enjoyed this biology lesson, and I look forward to seeing you next time. Thanks for watching. Bye.